In the grand, sprawling theatre of life on our planet, a constant drama unfolds. It is a story of survival, of continuation, of passing on the precious spark of existence from one generation to the next. Central to this narrative is the concept of relatedness, the intricate web of family ties that binds individuals together. We often speak of family trees, their branches reaching out, yet sometimes these branches can curve back, intertwining in ways that have profound consequences. This phenomenon, where closely related individuals reproduce, is known as inbreeding. It is not a rare occurrence confined to textbooks or laboratories. Rather, it is a thread woven through the history of countless species, including our own, across diverse landscapes and throughout the ages. Understanding it is key to understanding life itself. The idea of inbreeding might at first glance seem straightforward. It is simply mating between organisms that share a recent common ancestor, perhaps a pairing of cousins or even closer relations. This proximity in lineage means they carry a greater proportion of identical genetic material inherited from those shared forebears. Think of it as a family likeness, but on a much deeper molecular level. While shared traits can be a source of strength and identity, the concentration of genetic heritage through inbreeding also carries with it a hidden landscape of potential risks. These risks are not born of malice or intent, but are the simple, impartial outcomes of biological processes that have been shaping life for millennia, a stark reminder of the delicate balance that governs the health of populations. This exploration will take us on a journey into the heart of genetics. We will examine what inbreeding truly means at a biological level, peering into the microscopic world of genes and chromosomes. We will uncover the mechanisms that make it a perilous path for many species. A path that can lead to a weakening of vitality and a greater vulnerability to the challenges of existence. The natural world, in its immense wisdom, often provides clear examples, showing us both the instances of inbreeding and the strategies that have evolved to avoid it. These observations are not merely academic, they hold crucial lessons for us, particularly in an age where human activity increasingly impacts the genetic fabric of the species around us. We will also look into the annals of human history, where social structures, geographical isolation, or dynastic ambitions have sometimes led to patterns of inbreeding. The stories etched in royal lineages or remote communities offer compelling and sometimes cautionary tales about the consequences of limited gene pools. Finally, we will consider the broader implications for populations, how sustained inbreeding can diminish the vital genetic diversity that acts as a buffer against disease and environmental change. And importantly, we will touch upon the measures that can be taken and are being taken to safeguard this diversity, to ensure the resilience and continued flourishing of life in all its wondrous forms on this precious planet we call home. At the very core of every living organism, from the smallest microbe to the largest whale, lies a remarkable instruction manual. This is its genetic code, a blueprint written in the language of DNA. This code is organized into units called genes. Each gene provides the instructions for a specific trait, or a part of a trait, perhaps the color of a flower's petals, the pattern on a butterfly's wing, or the way an animal digests its food. For most complex organisms, including ourselves, these genes are inherited in pairs. One copy of each gene comes from the mother and the other comes from the father. This dual inheritance is fundamental to the diversity and resilience of life, creating unique combinations with every new generation. These gene pairs, however, are not always identical. Different versions of the same gene are called alleles. Imagine a recipe for a cake. One allele might be for a chocolate cake, another for a vanilla cake. Some alleles are dominant, meaning their trait will be expressed even if only one copy is present. Others are recessive. A recessive trait will only show itself if an individual inherits two copies of that particular recessive allele, one from each parent. If a dominant allele is present alongside a recessive one, the dominant trait will typically mask the effect of the recessive one. This interplay of dominant and recessive alleles is a constant dance within the genetic makeup of every individual. The closer the biological relationship between two individuals, the more similar their genetic blueprints will be. Siblings, for instance, share approximately half of their genes inherited from the same parental pool. Cousins share a smaller but still significant proportion. This genetic similarity is a natural consequence of shared ancestry. It's why family members often resemble one another in appearance, temperament, or even susceptibility to certain conditions. This shared inheritance is the very foundation upon which the concept of inbreeding rests, because when relatives reproduce, they are more likely to pass on identical alleles to their offspring, increasing the chances of those alleles pairing up in the next generation. Understanding this basic framework of inheritance is crucial before we delve deeper into the specific consequences of inbreeding. It is not about good or bad genes in an absolute sense, but rather about the combinations that arise. Life's genetic lottery means that every individual carries a unique assortment of alleles, some beneficial, some neutral, and some potentially harmful, if expressed. The system of inheriting one set of genes from each parent, especially when those parents are unrelated, generally promotes a healthy mix, a diverse portfolio of genetic instructions. This diversity is often a population's greatest strength, providing the raw material for adaptation and survival in an ever-changing world. When reproduction occurs between individuals who are closely related, the genetic lottery operates under a different set of probabilities. The key factor is the increased likelihood that the offspring will inherit two identical copies of an allele, not just from any source, but specifically from a common ancestor. This state of having two identical alleles for a particular gene is known as homozygosity. Conversely, having two different alleles for a gene is called heterozygosity. While all individuals have many homozygous gene pairs, inbreeding significantly elevates the proportion of these across the entire genome. It is a direct consequence of the parents sharing a considerable portion of their genetic makeup. Imagine a grandparent who carries a rare recessive allele, let's call it R. This grandparent passes it on to their children. If two of these children who are now siblings were to reproduce, there's a chance each could pass that same R allele to their own offspring. If their child inherits R from both the mother and the father, that child will be homozygous for RR, and the trait associated with this recessive allele will be expressed. Had the parents been unrelated, the chance of them both carrying that specific rare R allele and then both passing it on would be substantially lower. Inbreeding, therefore, doesn't create new harmful alleles, but it dramatically increases the chances of pre-existing ones meeting their identical twin. This concentration of genetic traits is not inherently negative for every single gene. If the alleles being made homozygous are beneficial, then inbreeding could, in theory, concentrate desirable characteristics. This is, in fact, the principle behind selective breeding in agriculture and for domestic animals, where breeders aim to fix certain traits like high milk yield in cows or a particular coat type in dogs. However, the genetic dice roll with every gene locus independently. While one might be aiming to make a beneficial trait homozygous, the process inadvertently also increases homozygosity for many other genes, including those that might carry hidden detrimental effects that were previously masked by a dominant, healthier allele. The degree of inbreeding, often measured by a coefficient of inbreeding, quantifies the probability that an individual has inherited two copies of an allele from a common ancestor. The higher the coefficient, the more homozygous the individual is likely to be, and the greater the potential for issues to arise. This is why the risks associated with inbreeding generally increase with the closeness of the relationship between the parents. A brother-sister mating, for example, carries a much higher risk of detrimental outcomes than a mating between second or third cousins, simply because siblings share a much larger fraction of their genes directly from the same two parents. The primary biological concern with inbreeding lies in its remarkable ability to unmask hidden genetic dangers. Most populations, whether animal or human, carry a reservoir of rare recessive alleles. Many of these alleles, if they were to be expressed, would be harmful, leading to genetic disorders, reduced fertility, compromised immune systems, or a general lack of vigor. In large, outbred populations, these harmful recessive alleles usually exist in a heterozygous state. This means an individual carries one copy of the harmful recessive allele and one copy of a normal dominant allele. The dominant allele functions correctly, effectively masking the presence of its potentially troublesome partner, and the individual remains healthy. However, the scenario changes dramatically with inbreeding. As we've seen, inbreeding increases homozygosity, the likelihood of an offspring inheriting two identical alleles for any given gene. When this happens for a harmful recessive allele, the individual inherits two copies of it, one from each related parent. With no dominant healthy allele present to mask its effects, the detrimental trait is expressed. Suddenly, a genetic problem that was silently carried and hidden for generations can manifest itself, often with serious consequences for the health and survival of the individual. This is the crux of what is often termed inbreeding depression. The range of conditions that can emerge is vast. They can include metabolic disorders, where the body cannot process certain substances correctly, leading to a buildup of toxins. Skeletal abnormalities, heart defects, and neurological problems are also commonly associated with the expression of harmful recessive alleles. Furthermore, inbreeding can weaken the immune system, making individuals more susceptible to infections and diseases that an outbred individual might easily overcome. It can also impact reproductive success, leading to smaller litter or clutch sizes, higher rates of infant mortality, and reduced fertility in the inbred individuals themselves, creating a challenging cycle. It's important to emphasize that these harmful recessive alleles are not created by inbreeding. They are naturally occurring mutations that are present at low frequencies in most populations. Outbreeding or mating between unrelated individuals acts as a natural safeguard, keeping the chances of these recessive alleles pairing up very low. Genetic diversity fostered by outbreeding ensures that even if an individual inherits one harmful recessive allele, it is highly probable they will also inherit a functional dominant allele from the other parent, thus maintaining health. Inbreeding effectively bypasses this crucial protective mechanism, bringing hidden genetic vulnerabilities out into the open with far greater frequency. Section 5 echoes in the wild nature's lessons on inbreeding. The natural world is replete with examples that illustrate the consequences of inbreeding. When populations become small or isolated, the choice of mates can become severely limited, leading inevitably to increased levels of relatedness between breeding pairs. Consider the cheetah, a magnificent predator of the African plains. Genetic studies have revealed that cheetahs today have remarkably low genetic diversity, suggesting they went through one or more population bottlenecks in their past, events that drastically reduced their numbers and likely forced periods of inbreeding. This genetic uniformity is thought to contribute to their high susceptibility to certain diseases and their relatively low reproductive success rates compared to other large cats. Island ecosystems often provide stark illustrations of inbreeding effects. Animals on remote islands may have descended from a very small number of founding individuals. With no new genetic input from mainland populations, generation after generation may involve mating between relatives. This can lead to the fixation of certain traits, some of which might be maladaptive. Scientists have observed higher incidences of congenital abnormalities, reduced litter sizes, and lower juvenile survival rates in various isolated island species, from birds to mammals. The kakapo, a flightless parrot from New Zealand, faced near extinction, with inbreeding contributing to very low fertility and hatching success before intensive conservation efforts began to manage its genetics carefully. Even in larger, more widespread populations, certain social structures or environmental pressures can lead to pockets of inbreeding. For instance, in some species with strong territorial behavior, if dispersal opportunities for young individuals are limited, they may end up breeding within their natal group, increasing relatedness. Royal albatrosses, for example, are long-lived and often mate for life. If their populations shrink, the pool of available unrelated mates also shrinks, and studies have shown that pairs with higher genetic similarity can experience lower hatching success. This demonstrates that even for species that are not typically considered inbred, a reduction in genetic options can quickly lead to negative consequences. These wild examples serve as powerful reminders. They show that inbreeding depression, the reduced biological fitness in a given population as a result of inbreeding, is not just a theoretical concept, but a tangible force that can impact survival and reproduction. Wild animals often show reduced ability to cope with environmental stresses, increased parasite loads, and a diminished capacity to adapt to changing conditions when their genetic diversity is compromised by inbreeding. Nature, in these instances, provides clear warning signs about the importance of maintaining a broad genetic base for the long-term health and resilience of any species. Section 6. A human tapestry. Inbreeding in our own history. The story of inbreeding is not confined to the animal kingdom. It is also woven into the rich and complex tapestry of human history. Throughout millennia, various human populations have experienced periods of inbreeding, sometimes due to geographical isolation, and at other times driven by cultural practices or social structures. Mountain valleys, remote islands, or expansive deserts have historically separated communities, limiting mate choice and leading to higher levels of consanguinity, marriage between close relatives over generations. In such scenarios, the genetic consequences observed in animal populations can also manifest in human groups, albeit with the added complexities of human culture and healthcare. Perhaps one of the most well-documented examples of sustained inbreeding in human history comes from European royal families. For centuries, dynasties such as the Habsburgs of Spain and Austria frequently arranged marriages between close relatives, including uncles and nieces or first cousins. The aim was political, to consolidate power, preserve wealth, and maintain the purity of their lineage. However, this practice led to a noticeable increase in certain physical traits and health problems. The Habsburg jaw, mandibular prognathism, became a prominent and heritable feature, and rates of infant mortality and genetic disorders were significantly higher in these families compared to the general population of the time. Beyond royalty, certain isolated founder populations have also provided insights into the effects of inbreeding. Small groups of people who migrate to a new area and remain reproductively isolated can experience what is known as the founder effect, where the genetic makeup of the entire subsequent population is heavily influenced by the genes of the few founders. If these founders, by chance, carried certain recessive alleles for genetic disorders, these alleles can become much more common in the descendant population. Communities like the Old Order Amish in the United States, or specific groups in parts of Finland or Quebec, have higher incidences of certain rare genetic conditions due to their historical isolation and patterns of marriage within the community. Understanding these historical and cultural contexts is vital. It is not about casting judgment, but about recognizing the biological realities. These human examples underscore the universal nature of genetic principles. While cultural factors shaped mating patterns, the underlying biological mechanisms remained the same. The increased homozygosity resulting from unions between relatives brought forth a higher incidence of recessive genetic conditions. Modern genetic counseling and increased mobility have, in many parts of the world, reduced the prevalence of such practices. But the historical record serves as a compelling human chapter in the broader story of inbreeding and its impact on population health. Section 7. The shrinking gene pool, long-term shadows on populations. The consequences of inbreeding extend far beyond the health of individual organisms. They cast long shadows on the viability and adaptability of entire populations over time. One of the most significant long-term impacts is the erosion of genetic diversity. Genetic diversity is the sum total of all the different genes and alleles within a population. It is, in essence, nature's toolbox, providing the raw material for evolution and adaptation. When inbreeding becomes common, the gene pool shrinks. Fewer variations of genes are present, and many individuals become genetically very similar to one another, a state of increased homogeneity. This loss of genetic diversity makes a population increasingly vulnerable. Imagine a diverse forest with many species of trees. If a new disease arrives that targets one specific tree species, the other species will likely survive, and the forest as a whole will endure. However, if the forest is a monoculture, consisting of only one type of tree, that same disease could devastate the entire ecosystem. Similarly, a genetically uniform population is at greater risk if a new pathogen emerges, or if environmental conditions change significantly. With limited genetic variation, there are fewer chances that some individuals will possess the necessary traits to survive and reproduce under the new pressures, potentially leading to a population crash or even extinction. This phenomenon, known as inbreeding depression at the population level, manifests as a general decline in fitness. This can include reduced average lifespan, lower overall reproductive rates, increased susceptibility to common ailments, and a diminished ability to cope with environmental stresses like drought, extreme temperatures, or food scarcity. Small, isolated populations are particularly susceptible because they often cannot escape the cycle of inbreeding once their numbers dwindle and their genetic diversity is lost. They can enter an extinction vortex where small population size leads to inbreeding and genetic drift, which in turn leads to lower reproduction and higher mortality, further reducing population size, and so on. Furthermore, the accumulation of mildly detrimental recessive alleles, even those not causing severe single gene disorders, can contribute to this overall decline in fitness. Many small, subtle negative effects can add up, making the population less robust, less resilient. The ability to respond to natural selection is hampered because the necessary genetic variation for selection to act upon is simply not there. In essence, inbreeding can rob a population of its evolutionary potential, its capacity to adapt and thrive in a constantly changing world. This makes the conservation of genetic diversity a paramount concern for the long-term survival of many species, including those managed by humans. Section 8. Nature's safeguards, instinctive wisdom against inbreeding. Life, in its relentless drive to persist and flourish, has evolved a remarkable array of strategies to avoid the pitfalls of inbreeding. These are not conscious decisions in the human sense, but rather behaviors and mechanisms, honed by natural selection over countless generations that serve to promote outbreeding and maintain genetic diversity. One of the most common and effective strategies is dispersal. In many animal species, particularly mammals and birds, one sex typically leaves the natal group or territory upon reaching maturity. Often, it is the males who disperse in mammals, while in birds, females are more commonly the ones to venture further afield before settling to breed. This dispersal effectively shuffles the genetic deck. By moving away from their birthplace and their close relatives, these individuals are far more likely to encounter and mate with unrelated partners. This reduces the chances of pairing with an individual who shares many of the same recessive alleles inherited from common ancestors. The drive to disperse can be incredibly strong, pushing young animals across challenging terrains and into unfamiliar territories, a testament to the powerful evolutionary advantage conferred by avoiding inbreeding. Even in plant species, mechanisms like wind or animal-aided pollen and seed dispersal serve a similar function, promoting genetic exchange over wider distances. Mate choice also plays a crucial role. Many animals have sophisticated ways of assessing the suitability of potential partners, and this often includes mechanisms to avoid mating with close kin. Scent is a powerful tool in this regard. Many mammals can recognize relatives through olfactory cues, such as pheromones present in urine or glandular secretions. Studies have shown that individuals may actively avoid mating with others whose scent indicates a high degree of genetic similarity. In some species, complex courtship rituals may also allow individuals to assess the vigor and genetic quality of a potential mate, indirectly selecting against those weakened by inbreeding. Furthermore, social structures within animal groups can inherently limit inbreeding. For example, in primate troops with clear dominance hierarchies, often only the dominant male or a few dominant males will sire most of the offspring. If these males are unrelated to the majority of the females, perhaps having immigrated into the group, this system promotes outbreeding. Conversely, if a dominant male's daughters mature within the group, mechanisms like their dispersal or the eventual replacement of the dominant male by an outsider prevent father-daughter pairings. These intricate dances of dispersal, mate choice, and social organization are nature's instinctive wisdom, beautifully evolved safeguards that protect the genetic integrity of populations. Section 9. Guardians of genetic health mitigating risks in a modern world. In our modern world, where human activities often fragment habitats and isolate populations, the natural safeguards against inbreeding can be compromised. This places a greater responsibility on us to act as guardians of genetic health, particularly for species whose survival is threatened or those under our direct management. Conservationists and scientists have developed a range of strategies to mitigate the risks of inbreeding and preserve precious genetic diversity. For endangered species, especially those with critically small populations, captive breeding programs in zoos and sanctuaries worldwide play a vital role. These programs often involve meticulous record-keeping, creating detailed stud books that trace the ancestry of every individual. These stud books are essential tools for genetic management. Coordinators of species survival plans, SSPs, or European Endangered Species Programs, EPs, use this information to make breeding recommendations. The goal is to pair individuals in a way that maximizes genetic diversity and minimizes inbreeding within the captive population. This might mean transporting an animal thousands of miles to mate with a genetically distinct individual. The aim is to maintain as much of the original genetic variation from the wild founders as possible, ensuring the captive population remains healthy and retains the potential for future reintroduction into its natural habitat should conditions allow. Molecular genetic tools are increasingly used to assess relatedness and guide these breeding decisions. For wild populations facing inbreeding due to isolation, genetic rescue can sometimes be an option. This involves carefully translocating individuals from a larger, healthier population into the small, inbred one to introduce new genes. A famous example is the Florida panther, whose numbers dwindled to a few dozen, suffering from severe inbreeding depression. The introduction of several female pumas from Texas in the 1990s brought new genetic material, significantly improving the health, survival, and reproductive rates of the Florida panther population. Such interventions require careful planning and risk assessment, but can be crucial for the recovery of critically endangered groups. Beyond wildlife conservation, practices in agriculture and livestock breeding also recognize the importance of managing genetic diversity. While selective breeding aims to fix desirable traits, breeders are also aware of the dangers of excessive inbreeding, which can lead to reduced productivity and increased susceptibility to disease in farm animals. Maintaining diverse genetic lines and employing strategic crossbreeding can help preserve vigor and adaptability in domestic species. Ultimately, whether in the wild or in managed settings, the principle remains the same. Genetic diversity is a cornerstone of health and resilience, and concerted efforts to prevent or mitigate inbreeding are crucial for the long-term well-being of the planet's biological heritage.